So in Unit 5.1 Part 2, I will talk about one of the groups of organisms that evolution has produced, the prokaryotes. So prokaryote is a term for any organism that lacks a nucleus, and it includes two distinct evolutionary groups, the bacteria and the archaea. Both of these groups of organisms are distinct in their biochemistry. However, they do function in some similar ways. Since they're single-celled organisms, they rely primarily on chemistry in order to survive in their environments, and they've been incredibly successful. So both of these single-celled prokaryotes live in all the parts of the planet, including on and in all other types of organisms on Earth. So prokaryotes are incredibly important for the environment. They play important roles in geological processes and in nutrient cycles on the planet. They photosynthesize and do all other kinds of important stuff, form the basis of food chains, etc. They're also really important for our health. And so in this class, we're going to focus on the impacts of prokaryotes on our health, both good and bad. So a habitat is an environment where an organism lives. Prokaryotes live everywhere on Earth. They live in the oceans, on the land, in the air, and under the soil. So the rhizosphere is the collection of microbes that lives on and in the roots of plants. And microbes that live on plant roots are incredibly important for nutrient cycles, to bring nutrients to those plants, to be primary producers for all the other organisms that eat food in the environment. We also have bacteria, then prokaryotes, that live in us, and we call this the human microbiota. This microbiota is the bacteria and other microbes that live on and in us at all times. We have at least as many bacteria cells in our body as we do human cells. And some estimates put that number as many as 10 times as many bacteria cells as human cells. We have both resident microbiota, which are microbes that live on us for long periods of time, and transient microbiota, which might change over short periods of time. You'll also hear talk about the microbiome. The microbiome is the collection of microbial genes in a person. So one of the important ways that we learn about the microbes that are there is by looking at their genes, as we'll discuss. And so the microbiome and microbiota are often used interchangeably, although they do specifically refer to two different parts of the organism. So prokaryotes live in communities. Even though they're single-celled organisms, they can interact both cooperatively and competitively with other organisms in their environment. So symbiosis is any interaction between different species in a community, and there are many different types of relationships that prokaryotes can have with other organisms. So our bodies are a prokaryotic community, and those microbes that live in us can have many different relationships. So most of those microbes as a population, they're living in us because they get some nutrients or some benefit from being in this relationship. Those relationships can include mutualism, where those microbes might be benefited but also give some benefit to us in living in or on us. We can have relationships called amenalism, where one organism is unaffected and another organism is harmed in the relationship. Commensal relationships are when one organism is unaffected by the relationship and another organism is benefited. So we might have many bacteria and microbes that live in us that really don't do us much good or bad one way or the other, but those microbes do have a warm place to live. Neutralism is when neither organism, even though they interact, has any net effect on each other. And then parasitism is a relationship where one organism is benefited and the other organism is harmed. And the pathogens that we'll talk about in this lecture and the next have parasitic relationships with us. So one important concept to understand when talking about prokaryotes is the concept of species is a little more complex than you might think. It is useful for us to talk about prokaryotic species, so groups of organisms that have similar characteristics. And it's useful to categorize, classify, and use that information, for example, to identify pathogenic bacteria. However, the traditional definition of species was developed for macro-level organisms, and that was that a species is a group of organisms that's capable of mating and producing viable offspring. But this doesn't work for prokaryotes because, like many other microbes, they reproduce asexually. And so this is a meaningless definition for bacteria. We can define species by their phenotypic similarity, so by the similar characteristics that we can observe in them. But the problem with this is that very closely related bacteria can have vastly different characteristics. For example, we have many commensal E. coli, which are non-pathogenic and live in our gut and don't cause us any harm. And we have highly pathogenic E. coli, such as the strain 015787, which cause serious intestinal diseases and sometimes death. We could also define a species by the genetic similarity of a group of organisms. But we know that prokaryotes especially are capable of exchanging genes through horizontal gene transfer. And we've seen many, many instances of this throughout evolution. So different species of organisms might exchange genetic information between their groups. So we do use the concept of species to talk about bacteria and archaea. But we also frequently categorize these organisms into more narrow groups, such as subspecies, strains, and serovars. And these are all groups of organisms that have some common characteristics based on different ways that we can measure those differences in similarities in the organisms. So OpenStax Chapter 4 does a good job of describing a lot of the different organisms, prokaryotic organisms that there are. I don't think that you need to know or memorize all of these different groups, but I am going to give you 4 specific examples from the most important groups that matter for human health. So Chapter 4 talks about the proteobacteria, which are gram-negative bacteria that include enteric bacteria. And enteric bacteria are bacteria that inhabit the enteric pathways of animals. So that is our small and large intestines. These include organisms such as E. coli, which I'll talk about. Proteobacteria are one large group of gram-negative bacteria. Other gram-negatives fall into several different groups. One example of a non-proteobacteria gram-negative is chlamydia, which we'll also talk about. Gram-positive bacteria are distinct evolutionary groups from the gram-negatives. Remember, they have a much different cell wall and cell membrane structure. They also fall into subgroups within the gram-positive. We have high GC% bacteria. Those are gram-positive bacteria that have a greater than 50% amount of G and C nucleotides in their genomes. These include the actinobacteria and specific species such as Mycobacterium tuberculosis, which we'll talk about. There are low GC% gram-positives, which have less than 50% G and C in their genomes, and these include Staphylococcus aureus. There are a couple of additional groups that you should know about, but don't have a huge impact on human health. For example, the deeply branching bacteria, which do not include any pathogens that we know of. These are extremophiles that are found in the harshest conditions on Earth. So these organisms live in some very harsh and isolated environments all over the planet. However, when we look at them, they are genetically similar even though they exist in physically isolated environments. These deeply branching bacteria are most similar, by our calculations, to the last universal common ancestor. The organism that presumably was the last common ancestor of all life on Earth. And so deeply branching bacteria are an important part of our understanding of how evolution has happened. We also should know about the archaea. So the archaea are a distinct domain from bacteria and eukaryotes. They are more closely related to eukaryotes evolutionarily, but they have many important biochemical differences from both bacteria and eukaryotes. They do live in a lot of the same areas that bacteria do, though. They make up, for example, about 1% of the human microbiome. Interestingly, though, we know of no archaea pathogens, even though the other two evolutionary groups, both bacteria and eukaryotes, are full of many pathogens. This seems to be an evolutionary strategy the archaea have avoided. We don't quite understand why, and it might be because we're only beginning to really understand the relationship, the importance of archaea in our health overall. So this is an active area of investigation. So what are some of these prokaryotes that you should know about? First is Escherichia coli, or E. coli, as it's known. It's a proteobacteria. It is a gram-negative bacilli. It's a facultative anaerobe, so it is able to live both in the presence of oxygen and in its absence. And it's famous for being a component of the intestinal microflora, the bacteria that live in the large intestine particularly, of many animals, including us. So there's about 0.1% of the human gut microbiota. So it is a significant but not a major component of our human microbiota. E. coli does illustrate some of the complex relationships we have with bacteria. So E. coli, commensal E. coli in our gut can have both mutualistic and parasitic relationships. So the E. coli in our large intestines play an important role in our health by synthesizing vitamin K, so that's a necessary nutrient for us. However, those same bacteria, if they get from the large intestines into your urinary tract, they can cause significant infections. And so same bacteria, depending on the situation, can be a good germ or it can be a bad germ. There are many different strains of E. coli, so subgroups with distinct properties. There is a famous laboratory strain of E. coli, which is one of the most well-studied organisms in all of biology, called K12. There are many different human commensal varieties or strains, and then there are many pathogenic strains, including the strain O157H7, which causes over 60,000 cases of severe gastrointestinal disease in the U.S. annually that result in about 3,000 deaths. So recently, many of these outbreaks have been linked to romaine lettuce. And the CDC particularly recommends that the romaine lettuce that is grown in the Salinas Valley of California should not be eaten because it has been a repeated source of pathogenic E. coli outbreaks. Another important prokaryote is Chlamydia trachomatis. So this is a gram-negative, but not a proteobacteria. What's interesting about chlamydia is that unlike most bacteria, but there are other examples of this in the bacteria, chlamydia is an obligate intracellular pathogen. And so it's only able to reproduce if it is infecting another host cell. The reason that chlamydia can't reproduce outside of the cell is it no longer can make, or the chlamydia cannot make its own ATP. All the ATP that chlamydia uses is absorbed from the host cell. And so it doesn't have any of that energy currency to drive many of its anabolic reactions unless it's inside the host. Chlamydia causes a number of infections. It is a sexually transmitted pathogen. and it has an interesting biology during those infections. In order to get into the cell, because it's an obligate intracellular pathogen, it goes through a number of structural morphological changes during this process. So the elementary bodies are the forms of the bacteria that are present outside the cell. So the elementary bodies aren't able to reproduce on their own, but they do induce the cells to phagocytize or absorb those bacteria into the host cell. Right. Once those bacteria are inside, they move themselves into vacuoles and then change into reticulocyte bodies. These are the cells that reproduce inside of the vacuole. Then they change back into elementary bodies to spread outside of the cell. Some good pictures down here of some of the diseases that are caused by chlamydia, including eye infection. So chlamydia is one of the major causes of blindness in the world. So Mycobacterium tuberculosis is a gram-positive organism. It's a gram-positive bacilli. It's in the actinobacteria, so it has more than, these are high GC organisms, so greater than 50% of its genome is G and C. It does not gram stain purple, even though it's a gram-positive bacteria. It is in a group of organisms we call atypical bacteria, because they don't respond like we expect them to the gram stain procedure. So instead, in order to diagnose MTB, we use a different biological stain called an acid-fast stain. And that image is shown there on the bottom. So this stains those bacteria red in using the acid-fast procedure. It's one of the most widespread pathogens. About 25% of the world population has been infected with Mycobacterium tuberculosis. It particularly causes a respiratory disease called tuberculosis. MTB is very slow growing, but even though it's slow growing, it is difficult to get rid of, and so it can cause long drawn out and long-term respiratory disease. Staphylococcus aureus is another gram-positive. It is a gram-positive cocci. It's a facultative anaerobe. It is a firmicute, and so it is in the group of organisms that are low GC gram-positives. Less than 50% of its genomes is G and C. It is a common part of human skin microbiota, and so about 30% of us have Staph aureus on our skin. There's a number of characteristics, phenotypic characteristics, including being salt tolerant or halotolerant. It produces an enzyme called coagulase, which allows it to protect itself inside of our bloodstream by coagulating human blood. It can cause a number of different important infections, including skin infections, soft tissue infections, food poisoning, bacteremia or infections of the blood, and also bone infections. So many Staphylococcus aureus are resistant to antibiotics, including a strain of Staph aureus called MRSA, or methicillin-resistant Staph aureus.